Chris Hague, thanks for joining MedTech Unboxed.
Steve, thanks for having me. Looking forward to the conversation.
Me as well. I'm really passionate about, as I mentioned to you, bioresorbables, and I was listening to your talk at LSI, and I was thinking back to the time when I first started to get involved or investigating bioresorbables and bioresorbable vascular stents. And in 2016, I held a conference, and my keynote speaker was James Oberhauser.
Hmm.
And I noticed on your recent LSI talk that he's part of your technical team.
I know Jim well. He's a great guy.
Okay. Yeah. He gave a great presentation in 2016 on the challenges with BVS designs and processing. So I think we've
Hmm.
come a long way from that time, right?
Well, if somebody would know, Jim's the guy. He's a
Yeah.
very talented, very knowledgeable guy in this specific field.
Yeah. So let's get into this specific field, and if you could tell us a little bit about Ephemeral Medical,
Hmm.
and then we'll talk about some of the the challenges in the peripheral
Hmm.
artery disease space.
Sure. Yeah. Well, Ephemeral, we're making a bioresorbable scaffold that's specifically designed from the beginning as a peripheral device. It's not adapted from a previous coronary design. And the challenges in the peripheral space are a little different from coronary stenting or coronary artery disease. So one of the differences with peripheral arterial disease is it tends to be in vessels that are larger. Vessels or the lesions tend to be longer. And very often, for reasons I think that are understandable, the disease tends to be a little bit more advanced when the patient presents for treatment. And you can imagine if somebody's got chest pain, they got coronary artery disease, it's pretty scary. They go to the hospital right away, they get treated. And coronary stents, as you probably know, are very effective, both in the short and long term. You get treated with a coronary drug-eluting modern stent, and you'll be fine for the rest of your life, pretty much. You probably
Yeah.
won't need another reintervention in that area. You might have progression of disease in other parts of your heart, but in that area where the stent is placed, they're very effective. And that's part of the challenge. I know you've had some discussion in one of your previous podcasts I was looking at this morning about the Absorb program from Abbott.
Hmm.
And the challenge for them that they ran into was that I think Absorb was a great product. It was developed and tested and proved worldwide. It actually provided great benefits to patients, but it just wasn't quite as good as the existing metal, either cobalt, chromium, or stainless steel stents. And as you know, the fundamental principle of device development is developing something for a large unmet clinical need. And I would argue coronary stenting was actually a clinical need that was met pretty well by existing technology. So
Hmm.
they kind of got tripped up by that. In contrast, for peripheral arteries, if you stent them or if you treat them with balloons, whether you have a coated stent or a coated balloon, the outcomes are just not very good. The failure rate is much higher, at the acutely, and the actual outcomes get worse over the life of the patient. So if you look at the failure rate in the first year, it's worse in the second year, it's worse in the third year, and it's just the disease in the leg, and the motion leg is a lot more complicated than the heart, and that's been the challenge for a long time, is how to develop a durable interventional device, whether it's a balloon, a drug-coated balloon, a stent, a drug-coated stent, atherectomy. Of course, there's now IVL lithotripsy for these devices. That's not really or for these cases. That's not really a destination therapy, but it's an enabling therapy that's very
Right.
important. So our goal is really to develop a bioresorbable scaffold that's specifically designed for peripheral arteries that will be durable over time.
You talked a lot about durability, and that's because of some of these failure rates, right, with predicates.
That's right. Yeah, I mean, just as a frame of reference,
Hmm.
if you implant, again, going back to the coronary versus peripheral
Yeah.
comparison, the failure rate for a coronary stent is 5% or less. It's very low over the life of the patient once it's implanted. And you think about it, the motion of the heart is just beating, right? So the stent doesn't have to move that much. It does have to withstand that pulsatile contraction of the heart. But if you think about that with the movement of the leg, when you sit down, when you bend your knee, when you walk, the skeletal motion of the main artery in your leg, called the superficial femoral artery, is quite complex.
Mm.
And the ability of a device that's able to keep that artery open over time is just much more challenging from an engineering standpoint.
Okay. Interesting. Yeah. Let's talk a little bit more about vascular scaffold system.
Hmm.
And one thing I wanted to know a little bit more, though, is some of the challenges with the predicates that might include a nitinol stent, which possibly doesn't have
Hmm.
enough strength, radial strength. If you can kind of touch on that a little bit.
Sure. All arteries, whether they're coronary arteries, peripheral arteries, they all actually prefer being stented by a balloon expandable stent. And what I mean by that is when you treat a vessel that has a lesion in it, usually, not always, but often those vessels have a lot of elasticity to them. They may have a lot of plaque burden, not just plaque, but calcium in the plaque burden. And so they're quite resistant often to expansion. So with a balloon expandable stent, you can really get a lot of radial force, with the delivery system itself and the stent itself to be able to really expand the vessel and treat the lesion fully. So if you think about with a coronary intervention, if you have a stenosis that's maybe at 80 or 90 or 95% blockage, when you put a balloon expandable stent in there, you get the blockage down to 5% or less. It becomes very low, almost, fully restoring the original lumen of the vessel. With a nitinol stent, if you're using it in the leg, of course, the importance is you have to have a device that's not going to break or fracture when the body is in motion. This is why nitinol is used, is you can put nitinol in a vessel like the superficial femoral artery or a tibial artery below the knee. And when you bend your leg and move, it'll flex with your vessel or with your body. But it doesn't have a lot of radial resistive force. So the actual forces of being able to restore the lumen to its full diameter are limited. And if you look at the historical literature, as I quoted, let's say a 5% or less residual stenosis with a balloon expandable stent in the heart, whether it's a self-expanding stent or whether it's a balloon, the residual stenosis with peripheral interventions tends to be 20 to 25%, often as high as 30 maybe. And in that case, once the stent is there, it doesn't take much neointimal growth or tissue proliferation to get to 50%, and now you're at restenosis. And so that's been part of the challenge previously, is just getting the vessel completely open at the index procedure. Of course, you want it to stay open over time, but you have to fully restore the lumen at its initial procedure. And the radial resistive force of nitinol is just limited. And if you hold it in your hands, I know you're a stent expert because I've watched some of your previous podcasts. But if you hold a balloon expandable stent in your hand versus a self-expanding stent, they just feel very different.
Yeah.
The other challenge is that with a self-expanding nitinol stent, there's a continuous outward force against the vessel. This is how those stents are anchored in the vessel. They're usually sized slightly larger than the reference vessel. That way they don't migrate.
Yes.
But the problem with that is there's always this interaction between the stent and your artery, which stimulates sort of tissue growth, irritates the vessel, so the vessel can't fully heal in that context. Whereas with a balloon expandable stent, you place it in, there's a transient injury at the time of expansion, but then the stent is there, it stabilizes the artery, it allows it to heal, and then if you've got a anti-proliferative drug on it, you can prevent the over-aggressive scarring response known as restenosis that may happen afterwards. So that's the ideal. This is why coronary stents work so well, is they're used with balloon expandable delivery systems.
Sounds like the nitinol, wonderful material, right? And because of several reasons, one of which the super elasticity, but in this case, it comes at the expense of radial strength, and it's what I'm kind of hearing. Yeah.
Exactly.
Okay. Interesting.
Yeah.
You talked about tissue growth, just to cover that before, and is that related to endothelialization?
Yes. So what you want is when you place a stent, whether it's a nitinol stent or a cobalt chromium stent, whether it's balloon expandable or self-expanding, you do want the vessel to heal around the stent, and what you want is a endothelial layer to grow over the struts of the stent, so the struts are not exposed to blood as it's flowing in the artery. That's what you want for a healthy sort of healing response. What happens sometimes, unfortunately, is that that response becomes a little bit over-aggressive. The healing response, you get a lot of a smooth muscle cell migration that comes through the struts of the stent, and then if it happens too much, you actually get sort of a reduction in the lumen of where the blood can flow, and then you get into what's known as restenosis, where you've just got too much tissue growth, kind of like scar formation inside the stent. So you want a layer of endothelium to cover the stent, but you don't want so much to occlude the blood flow.
Okay, excellent. Thanks for sharing that. I was curious about that particular one there. I was going to share my screen if you'd like, and you can go
Sure.
over the ephemeral vascular scaffold system, which is
Happy to.
fascinating to me, and I'm sure it will
Okay,
be
great.
with others. Yeah.
Great. Okay. So, yeah, so here's a schematic of the ephemeral vascular scaffold system. What you see is it's a balloon expandable delivery system on top of which we crimp a series of independent bioresorbable elements. These are unconnected scaffolds.
Mhm.
And in their crimp state, it looks like one continuous device, but in fact, in this embodiment, there are five. You can put fewer, you can put more on, so you can have a shorter or longer device depending on what the needs are for any particular patient. In this case, there are five scaffolds. This produces a 60 millimeter long device. And what happens is when you inflate the delivery system, these scaffolds expand on top of the delivery system, and by design, they foreshorten slightly and create these interscaffold spaces. So the key design input here, Steve, is when we talk to our stent engineers about how to design these individual scaffolds, the emphasis was all around radial strength. And one of the ways you make a device strong, a scaffold strong, I should say, is through what's known as a closed cell design. So you can see those are individual kind of diamond shaped patterns.
Yes.
This allows them to be very strong because each individual scaffold does not need to be flexible because of those interscaffold spaces. And if you move to the next slide, I think you'll see what I'm talking about. When you remove the delivery system, you see there on the left, the system overall, the scaffolds remain in the artery, but they are able to move around the artery because of the interscaffold spaces. But each individual scaffold is quite rigid. And
Hmm.
so it's all about radial strength. We achieve that through that stent pattern that I mentioned. We also
Yes.
achieve it through the strut thickness of the device, as well as the polymer orientation. And speaking of polymers, we are also made of PLLA with a copolymer.
Yeah.
In fact, back to your reference of Jim earlier in the podcast, Jim was very nice of us to work with us and suggest that we put in this copolymer because it does a couple of different things. Rather than being 100% PLA, which is quite brittle sometimes,
Mm.
if you remember
Yes.
some of the early experience with Absorb, you had to do very careful work in prepping the vessel. You had to be very careful not to overinflate the scaffold so it wouldn't break. You had to kind of baby it a little bit to get it to work properly. But with a copolymer, the scaffold becomes much more ductile, much more, let's say, less prone to fracture. In fact, we
Yes.
haven't seen any fracture in our early animal studies so far. The other benefit that we thought might be the case, and in fact, we do have evidence now, is that rather than taking 4 to 5 years to fully resorb, which is the case with 100% PLA design, ours is fully resorbed by the body somewhere between 1 and 2 years.
Okay.
So the actual function of the scaffold is pretty much complete by about 6 months after implantation. And so you want it to be strong and eluting drug over that period of time. But once it's done its job and the vessel is healed, you don't really need the scaffold anymore. And so you'd
Right.
rather it go away, earlier rather than later. Um, and then the third component here is we do have a sirolimus coating. Sirolimus is a well-known antiproliferative drug that's been used on coronary stents for many years. And we have a version of that, which is a relatively high dose and a relatively prolonged period of elution.
That's excellent. On the flex step, not only with the segments and the spacing, I would assume that that particular platform design would also lend itself well for deliverability.
Yes, it
Compared.
does. In fact, one of the other advantages of having this segmented design, this flex step, is we can make a very long device, right? So all you have to do is just have a longer balloon and load more scaffolds onto it. And so with a continuous, let's say, scaffold or a continuous stent, there is going to be a limit of how long you
Mm.
can make that. Of course, nitinol, you can make them quite long. So I don't want to give the impression you can't have long nitinol stents, but of course, then you get back into that, lack of radial strength and chronic
Yeah.
outward force on the vessel. So this
Right.
is a way to make a very rigid stent quite long as well. So this is how we sort of solve for that issue of radial strength, but also the ability to make it very long. And long scaffolds are important because that's really where most lesions are. Most lesions are not short, kind of narrow chip shots. Most lesions are quite complex, often quite long, often 100, more than 100 millimeters, up to 200 millimeters. Some people even longer.
Yeah.
And this segmented design allows us to make a truly long device, which is going to be clinically quite applicable for the physicians who are treating these patients.
Okay. Wow. That's excellent. Thanks for sharing that.
Steve, I'm
What,
gonna, if I may, I've just got a little. I don't know if this is going to show on your on my camera here, but this is one of
yeah.
the individual scaffolds. If I put it up there, I don't know if you can see that very well. If that's focusing or not, but that's what our individual scaffold looks
Ah,
like.
okay.
And
One of the segments.
each one is like the, yeah, each segment is like the one next to it. So there's no difference in terms of the first or second or third scaffold. They're all the same.
Okay. You talked about peripheral arteries in the leg having more space and they're longer. And I always noticed when I look at just the balloons, coronary versus peripheral, just they're so much longer. And you kind
That's
of
right.
explained that a little bit about it's not that it's advantageous to have a longer. It can be a longer stent as well, stent segments with the balloon.
Yeah, absolutely. And one of the things that I may have skipped over when I was talking a little bit earlier about the difference between coronary and
Mm,
peripheral
yeah,
disease is if you have disease in your leg, let's say some narrowing in your leg, it's causing claudication. A lot of people kind of shake that off. They stop walking. They kind of wait for they kind of collect themselves, and they said, "Oh, I'll go to the doctor later when I have time," or "It's not a big deal. I'll just
yeah,
kind of I'll grit it out." And so by the time these patients end up not being able to ignore it anymore, the disease tends to be quite advanced by the time they end up on the table for treatment. So that's another key difference between
yeah,
coronary disease because patients go right away to the doctor,
yeah,
whereas peripheral disease, the patients tend to put it off a little bit more.
yeah. Interesting, interesting. Excellent. Thanks for going through the kind of the unmet needs and your scaffold design. Very, very interesting. I wanted to see if you can give us an update on clinicals and
Hmm.
and then maybe an update on any funding.
Yeah, so we are in the middle of conducting a first-in-human trial called Ephemeral 1. This is a single-arm study looking at patients who have claudication, which is that condition of a debilitating pain in the leg or buttocks area. It's a multi-center study. We're at 4 sites across New Zealand and Australia. These are very well-known researchers that we're working with. You probably know from your conversations with other entrepreneurs that New Zealand and Australia have become a place that many people commonly go to for their first-in-human trials, and
I've
that's
noticed. Yeah.
we're similar with that.
Okay.
And to date, we have enrolled 33 patients in Ephemeral 1. These patients are a different stage of follow-up because, of course, what we're looking at is not just how does the procedure go? Is it uncomplicated? Is it a safe procedure? Want to make sure we don't have any thrombosis, acutely. But we also want to follow these patients over time. So we have a subset of these patients that are being looked at very closely at 6 months. So with an angiogram, also with OCT, which is optical coherence tomography, and some other imaging modalities, CT, MR, CT, and so on, because we really want to look in these early patients how these vessels are doing, not only from the outside, but also from the inside. And then all the patients get follow-up with duplex ultrasound at key time points at 6 months, at a year, 2 and 3 years. So our earliest patients have now had their 3-year follow-up. We've had a few more patients that have their 2-year follow-up, and quite a few now that have had their 1-year follow-up. Now, the results from the first 20 patients will be presented later this month on April 23rd by Dr. Andrew Holden, who's our principal investigator. So I do want to respect the confidentiality of
Yeah.
what those results will be. This will be at the Charing Cross Conference in London on April 23rd. But I can tell you that I think they're going to open a lot of people's eyes as what's possible with actually treating this disease. This disease, as I mentioned, has been not very durable in terms of its outcomes of how you treat them over time. In fact, you know, as you probably know, Steve, is in the leg, unlike the heart, a lot of people try to avoid using stents just because they
Yeah.
can cause complications, and the outcomes are not that great.
Right.
But I think for the first time, people are going to see a device that actually can provide durable outcomes, not just good acute outcomes, but also durable outcomes. And but I'll leave it to Dr. Holden to discuss that for the first time, the details, and then of course we'll put out a press release around that when the time comes.
Yeah, and I'll be sure to share that press release. I'll put a note in my
Hmm.
calendar for April 23rd. Looking forward to that. We don't want to.
We're looking forward
Yeah,
to that too.
yeah.
Yeah.
We don't want to get ahead of ourselves, so
Hmm.
we'll keep up to
Yeah.
date on that.
Yeah.
Excellent. And then just lastly, any news on funding that you'd like to share?
Yeah. Yeah, we have, we're fully funded through basically angel investors so far. We've raised a little
Okay.
over 10 million in equity so far. We've also raised an additional little bit over 2 million in terms of non-dilutive SBIR grants. We got a phase 1 and a phase 2 grant. And that has allowed us to meet our original milestone for ourselves was to get to 30 patients enrolled in our first in human trial that I mentioned just a moment ago, the Ephemeral 1. And we've already kind of passed that. We're going to keep
Yeah.
going. We're still enrolling patients. So I think that number will climb as we progress here. But we are looking to raise a Series B right now. We just kind of opened the gates and speaking with investors. We were actually at the LSI conference that you referred to a little bit earlier, speaking with investors. And the Series B, the funding there will go to all of the work we have to do to get to a high quality ID submission. As you know, we're a PMA path device, so we have we have to do all the different device testing and a randomized trial later on, to get approval here in the U.S., which is our key goal, is U.S. approval, of course.
Yep.
But for an ID submission, we have a series of animal studies, GLP animal studies we'll need to conduct. We've already had a Q-sub with FDA, so we've negotiated what those studies look like, how many pigs we're going to do, what time points we're going to do, sample sizes, all that. That's all part of our budget. The other piece is we have to build and test a bunch of devices, all the validation testing, the verification testing, the drug stability, the biocom testing. That's all part of the package. And then any additional trial enrollment we think might be necessary to get to that high quality ID submission. So that the Series B, we're looking to raise it. It's in the range of $20 million. That's going to last us about 2 years.
Okay.
I think we can actually be a little bit more efficient than that. Even we've so far we've been very efficient. I mean, on the money we've raised, we've already got 33 patients. I think that's pretty efficient compared to maybe some other companies you've talked to in terms of how much it costs to get to this point. And we plan to continue our sort of ways of being efficient and good stewards of our investors' money. And then once we get the ID approval, then of course we'll have a Series C, which we call a clinical round, which will fund the pivotal study. It'll come in a couple of years.
Okay. Are you for your DV builds? Are you kind of doing that yourself? Are you working with a manufacturer? Or you don't have to share that if you don't want. I was just curious.
We have, currently the way that we've been able to make so much progress with a modest investment so far is we are working with some very knowledgeable and experienced third-party vendors. We get components from these vendors, but we tend to do the final assembly ourselves.
Okay.
So we take those components from our various vendors and we give them kind of a formulation, a recipe of what we need from them, in terms of the tubing, the coating, there's our devices also laser cut.
Yeah.
We have, of course, a balloon delivery system that we get, but we take all those components and we have a crimper that we've had custom made for our device, and we have a particular crimping recipe that we use because we want to make sure that we don't damage the scaffolds during the process of crimping. And then we do that final assembly, send it out for sterilization, and then we do the testing.
Okay, thanks for sharing that. Interesting.
Mm-hmm.
Chris, this has been a real interesting conversation. I look forward to April 23rd to get more results. Yeah, and
stay
and
tuned.
your
I
series
think we'll open a lot of eyes.
Yeah, and that'll open hopefully the doors to your Series B as well, right?
I hope so. I mean, we've had a lot of good conversations. One of the things that from your previous podcasts is there's been a lot of research and interest in bioresorbables. Abbott's done some great work with their Esprit BTK device. Yes. There are a few other companies out there that have raised money recently with a similar device. I think we're differentiated in that we have a tibial device, but we also have an SFA device. We can make a very long stent, or scaffold, I should say.
Yeah.
So we won't be the first, but I think that we have some unique advantages that would be to our benefit.
Okay. Well, I'll look forward to following you.
All right. Well, Steve, thanks for the opportunity, and I will. Hopefully, you'll see the results on the 23rd, and maybe we'll have a follow-up conversation.
Absolutely. Thanks, Chris.